In this video, we will discuss about some of the results that shows that HTPBD has excellent tumor enrichment ability and has high transfection efficiencies compared to its counterparts. So in the panel A, they check the diffusion ability of PBD, HTPBD and HTPBD, and what they are able to find out is that HTPBD has more tumor penetration ability compared to the PBD and HPBD. And this is the graphical representations is shown that HTPBD can penetrate within this mucus and can target the tumor cells. And in the panel B, it shows the results in which the diffusivity of this plasmids PBD, HPBD and HTPBD in the mucin solution is shown there. And we can see that HTPBDs after 6 to 12 minutes, we can see the difference in the results for the HTPBD diffusivity and PBD and HPD diffusivity, and these things they have quantified in the panel C, where we can see that the diffusion coefficients of the HTPBDs are significantly higher compared to the PBD and HPBD. And they did a transfer assay for these CACO2 cells, and they are able to see that in the CACO2 cells the permeabilities in the CACO2 cells, they are able to see that the diffusivities are higher in the CACO2 cells also, and these things they are quantified in the panel E, where we can see that in the control case the diffusivity of all the systems are almost similar, not similar. This HPBD and PBDs has the higher transfection efficiencies compared to the control, that is 31.5 compared to the 1.88, whereas HTPBD has transfection efficiencies in the PBS around 50.5. But when they use the mucin, they can see that in the mucin side they can see that the transfer through the transfer assay they can see the diffusion probabilities of these HTPBDs are higher, that is around 28.1% compared to the control case, that is HPBD and PBD, that is 6.5 and 4% respectively. And then they check the permeabilities through these CACO2 cells, and they are able to see that the permeabilities through these CACO2 cells or trans viscosity, I'm not able to recall the exact term. It means that how easily their reagents or the vectors that they have generated are able to pass through these CACO2 cells, and they can see that the HTPBDs can has higher permeabilities, that is around 28.5% compared to its counterpart that has around 7.85 and 7.66%. And the reason why they choose this CACO2 cells is because the CACO2 cells has this octi-N2 which can interact with this T part in the HTPBD, which is TMAO. So it can interact with the TMAO and increase the diffusivities of the this HTPBD. And these things, in order to validate this thing, what they did is they check these CACO cells. They check these CACO cells permeabilities using a different system, that is PBD, HPBD and HTPBD, and then they inhibit this T-N2 using cimetidine and L-carnitine, and they can see the reductions in the diffusivities of these HTPBDs from 33.3% to 18.3 and 19.2%. And the immunofluorescence image are also shown here, where we can see that with these HTPBDs, PBD and HPBD, the HTPBD signals, the plasmid signals are higher, and when they use the cimetidine and L-carnitine, the signals goes down. However, when they did the similar studies in the control cells, they do not see any changes. That is, HTPBDs has around 11% this diffusivities, and with this cimetidine and L-carnitine they do not see a significant changes, indicating that it's the octi-N2 that is causing that is helping in the increased diffusivities of these HTPBDs. Then they did some in vivo studies where they find out that with these HTPBDs they are able to see more penetrations and that can last for a longer duration of time compared to the PBD and HTPBD. And these things they are quantified in the panel M, where we can see that even after 12 hours and 24 hours they are able to see the fluorescence intensities almost higher and compared to the PBD and HPBD cases. And they did their studies on the tissues also, in vitro studies on the tissues also, and they find the similar results, that for the HTPBDs they see a higher fluorescence intensities compared to the HPD, HPBD and PBD. And these things parallel ends, the results are being quantified in the panel N, where we can see that in the case of tumors only they can see the difference in the intensities, fluorescence intensities, whereas for the heart, liver, spleen, lung, kidney they do not see any significant changes in the fluorescence intensity. This shows the specificities of their HTPBDs that can target the tumor sites but not the other sites, that is heart, liver, spleen, lungs, which are not affected by tumors. So their results in all together shows that this HTPBDs has some excellent tumor enrichment abilities and high efficiencies compared to their counterpart, that is PBD and HPBD, for this mucin related tumors. If you need to understand more about their experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.